Okay, so let's say that investigator Y is interested in looking at a virus in CFS. We know that the best way to look for viruses is to collect them in a tube that allows the blood cells in the tube to be able to be isolated and then grown up in tissue culture. And there's 2 tubes that can be used for this type of purpose. A lot of people that have tried to isolate and characterize viruses have used the different types of tubes. So the nice thing about the biobank is that we can say, okay, what is it that you need in order to do your experiments? And then we can customize it exactly that way. So if you are a participant in the biobank and you consent to us collecting your clinical information and providing samples, we call you up and say, we have a study, we need this type of tube, are you willing to give it? So essentially what we've done now is create a bank that is customized to the needs of the individual investigators. Now the other nice thing about that is it's not a one-shot deal. So investigator Y has found a virus, and now another investigator wants to validate that. Because we have a bank of samples that were collected in the exact same way, that investigator can go back to those samples or an expanded set and see whether or not they can reproduce it. Okay? So the biobank is a remarkable resource that I think is going to, instead of a 20-year time frame for biomarker identification, reproducing, and validation, it now shrinks it down to 5 years. 3 years for me, but it shrinks our time frame down because we are now providing what was a non-existent entity as a resource to the research community. And the timing for this couldn't have been better with the finding of XMRV. Okay, so Cindy tells me I have 1 minute. So do all of you understand what a biobank is? Okay, and again, I encourage you to get the brochure that, that my organization shipped out here. And just a show of hands, how many of you would be interested in participating or being a member of the biobank? It doesn't cost you anything. Okay, super. For those of you who didn't raise your hand, come and see me later. Yeah. Real quick update on the first International XMRV workshop that happened in Washington at NIH, National Institutes of Health, on Tuesday and Wednesday. It was a jam-packed meeting with a lot of information covering the basics of XMRV and polytropic MLV retrovirology. We also had talks on XMRV and prostate cancer, and we had talks on XMRV and polytropic MLVs and CFS. The take-home messages from this meeting are up here. There's no conclusions, okay? We're really early in this. There are no conclusions. I think there's just a number of very important take-home messages for the scientists and the MDs and pharma that participated in this meeting. First take-home message, these viruses I have up here, these viruses. I should fix that and say polytropic MLVs, which are polytropic murine leukemia virus, mouse viruses, are common, and they're very widespread among house mice and lab mice. And it turns out many other mammalian species. So cats got them, dogs probably got them, mice got them, okay? So these murine leukemia viruses are common. Now, cats and dogs don't have murine leukemia viruses, they have their own, but they're very closely related. So far, infectious virus from polytropic murine leukemia viruses has not been demonstrated. And the reason that these polytropic murine leukemia viruses are of interest is because a paper published by the NIH and the FDA and proceedings of the National Academy of Sciences last month showed these polytropic MLVs in 80% of CFS patients. It was a relatively small group of CFS patients, but 80% nonetheless. They were not able to find the infectious virus, like the Science paper found infectious XMRV in CFS in Science back in 2009. Also, an important take-home message is that XMRV is not the same as polytropic MLV. These are different viruses. Similar, related, similar in the way they look. Dr. Singh showed a picture of that this morning. Somewhat genetically related, but they're different viruses. There's discrepancies in what the investigators are finding, whether it be in prostate cancer or in CFS. Some investigators find XMRV and or poly MLVs, and others don't. And that the reasons for these discrepancies could be the different patient samples that they've studied, the different techniques that they've used, and the different samples that they've tested. However, there is great excitement and opportunity for research now. CFS research in particular. The director of NIH, Dr. Collins, was at this meeting almost the entire time, 75% of the time. I have never seen this in my life. To have the director sit in the front row and listen to a series of talks on XMRV and polytropic MLVs. He even asked questions. I mean, it was quite remarkable. The Red Cross and other blood banking communities are interested in what's going on with XMRV and polytropic MLVs. Pharma's interested in these viruses. CDC is interested in these viruses. FDA and the academic community. This is a phenomenal change in this area now. The way I look at it, can you guys see my little XMRV there? XMRV has put CFS on the radar, okay? And now, I think by engaging investigators that have not been engaged in CFS research before, it's going to be very different, and by not only investigators, but all these communities, the blood community, pharma. I don't think it's going to be easy for CFS to fall off the radar map. Already, there are a number of studies underway because of XMRV in CFS. The first one being to determine how common XMRV is in populations of CFS. This is a classic prevalence study. We don't know who's going to do this yet, but I think there's already a number of players vying for this particular study. There's the CDC, there's the Red Cross, there's the blood community in general, because it's important to know that if there's an infectious agent in CFS, then it could, in the blood, it could be a threat, a possible threat to the safety of the blood supply. And obviously, it has implications for people with CFS that have the virus. There is also a lot of discussion, and studies are now being thought about once we determine how common the virus is, and once the tools are developed to reproducibly detect XMRV, whether or not we can identify those individuals and then bring them in for clinical trials. And that's clinical trials with antiretrovirals. Because as Dr. Singh showed this morning, we do know that there are some antiretrovirals that have effect against XMRV, or that are efficacious in treating or decreasing the replication of this virus. Another big question that's currently being investigated is whether or not XMRV is transmitted in the blood. And again, we have questions about donation. How many of you in this room donate blood? Or have donated blood in the past 10 years? Past 10 years, okay. There's also transfusion studies that are beginning to startup form. How many of you have received the transfusion either before or after you got sick with CFS? That's not as many, but clearly still quite a few. Please, for those of you who have donated blood and/or transfusion, we're working with the Red Cross to do what's called a look-back study. Okay, so if we can, if you are interested in participating in such a study, that doesn't necessarily look directly for XMRV, but it will say whether or not CFS is transmissible, whether it be by XMRV or some other agent. And there are a number of developing animal models that are ongoing. Okay, so I think that's all I have.